How can multiomics and deep immune profiling approaches help disentangle the contributions of sex, age, and environment to variable immunotherapy responses?
Yeah, so we will need to know exactly how these factors affect the genome and the epigenome, right? And so we have some information, but um, we don't know exactly all the pathway. What I can tell you though is that there are studies now that look at how diets, for example, exercise impact the transcriptomics. So we're starting to have signature that can predict being a surrogate marker of how the patient is exercising, how the patient, what type of diet. For example, think about the um, metabolomics on stools, metabolomics on serum. Um, just the study of the microbiome. These are all factors that we can measure, and we can get a sense of the quality, for example, of the microbiome, and we know that has a huge impact. So once we're gonna learn more about the exact pathways, we're gonna be able maybe to say, well, instead of just changing your diet, maybe you can get these two supplements that are gonna do exactly the same thing. We're not there yet, though.
From a translational standpoint, what are some concrete strategies being proposed to tailor immunotherapy, such as dosing, combinations, or sequencing based on host factors? Are there?
Yeah, I don't think we're yet there. I can tell you a little bit about what we are doing, for example, in the setting of diet. So we last year, actually, at the plenary session here at the American Society of Hematology, we presented a study where we showed that ketogenic diet is actually a very good diet to improve CAR T cell function in preclinical models. Thanks to that work, we are now studied a trial at the University of Pennsylvania where we're giving beta hydroxybutyrate, which is a ketone body that is enriched in ketogenic diet, and we're measuring the effect on the T cell function. And of course, we're also measuring the clinical outcome. So this is an example that tells you that if we understand the basic mechanism, for example, what are the metabolites that T cell need to function? We give the example of the ketogenic diet and ketone bodies. Well, we might provide them to the patient to improve the outcome. That's one example. Other groups, for example, are doing fecal material transfer. So they're basically doing a transplant of the fecal material to improve the microbiome and improve outcomes. And for example, this has been shown to be effective in the setting of checkpoint inhibition. So again, you can see that we're already sort of informing some of the strategies, but I mean, none of these approaches are by all means routine or FDA approved.
Mhm.